Welcome to Georgia College's fabulous Barrie campus. We're here recording live in room A120. My name is Scott Drouet and I have an anxiety disorder. This is my associate Brandon Curtin. Today we're going to talk to you about biological pathogens that can affect freshwater fish in Ontario's lake ecosystems and the potential damage that these diseases could do to food webs.
Now, some of these pathogens could include viral hemorrhagic septicaemia or coldwater disease. Now, Scott's going to explain to you a few of the other details of some of these pathogens.
That's right. Now, viral hemorrhagic septicemia was only recently introduced to Ontario's Great Lakes in 2002. It's a disease that affects teleosts. Now, you may be wondering, does this guy know the definition of the word teleost? In fact, I do. A teleost refers to a diverse taxonomic group of fish with bony skeletons and includes most of the fish species on Earth. Now, when I say bony skeletons, I mean that this group is differentiated from cartilaginous fishes such as sharks, rays, and skates. Now, there are 4 distinct genotypes of the VHS virus. And again, you may be asking, does Scott really know what the word genotype means? In fact, I do. The word genotype simply refers to the specific genetic makeup of an organism. So that means that there are 4 distinct genetic versions of the VHS virus. But wait a minute, in 2002, a new genotype was discovered in the Great Lakes and caused a large number of mortalities in a variety of teleost fish species.
We're back. We're now going to have a little chat amongst the two of us regarding viruses and their effects on food webs and potential transport of the viruses.
That's right, a simple exchange of ideas. Now, Brandon, when I think about the transport of pathogens that affect fish, I think that they're characterized mostly by the migration of fish. I mean, if you have infected fish in one lake, and that lake is connected to another lake by a stream, then those fish could potentially swim up the stream and infect other fish.
Good point, Scott. Now, you also got to think about the fact that interwaterway transportation of humans using their boats in waterways such as the St. Lawrence or the Trent River system.
Well, no doubt. In fact, that's another key risk factor in infecting new lakes with viruses and pathogens, such as the viral hemorrhagic septicemia virus. Now, anthropogenic transport of live and frozen fish is quite common and often goes untested or undertested. And one of the studies I read actually showed that viral infections are not always killed by freezing the fish. So frozen feed products for aquaculture could potentially contain diseases such as viral hemorrhagic septicemia.
Interesting.
Also, the transfer of live fish for reasons such as restocking of and bolstering populations that are unstable.
Like Lake Simcoe?
Exactly. In fact, I hear that a large number of both whitefish and lake trout are introduced to Lake Simcoe every spring.
Yeah, I heard that before as well.
In situations like that, there's also the risk of exchanging bacterial pathogens, that is, from the densely populated scenario where these fish are bred and raised, and then introducing it to the lake. Diseases such as the coldwater disease of fish, which is known also to be active in Ontario.
Interesting. Now, do you think that introducing some of these fish with some of these pathogens into an already natural system could affect the food web in the system?
I most definitely do. In fact, I think, especially in lower connected food webs, and especially situations maybe where there's less interconnectivity of lakes, if you can introduce that pathogen there, you're going to have a weaker, less complex food web, where damaging one species in it could really affect the overall ecosystem.
That's right. Actually, some of the research that I did, Scott, gave us some visuals that we could use to show people how losing some of the trophic links due to pathogens could ultimately lead to the decimation of the food web.
Oh, like this example here, you mean?
As you can see in this first example, if we were to lose the trophic links between these 3
isolated clusters,
we could end up with 3 separate splintered food webs instead of one continuous food web, as in this example B, which has a little bit better connectivity.
That makes sense. And this isolated species here looks like it's on the way to extirpation.
Most likely.
Now, I would, if I hadn't already done research on this, wonder how an isolated food web could be exposed to a virus such as this. Well, we've already talked about the exchange of live bait fish, feed fish, frozen fish. What I haven't mentioned is something called fomites, which is actually any substance that's capable of carrying or transmitting biological pathogens. So things like this could be fishing equipment used in an infected lake, boats, it could be waste from fish processing plants, and another thing that I haven't mentioned yet could be the migration and transport of piscivorous birds that could excrete these pathogens in their feces.
Good point. Birds such as, I don't know, ducks and geese and maybe cormorants?
Definitely cormorants, as we know that they are piscivorous and migratory.
Interesting.
Well, when I think about aquaculture, I think about, in many of the studies of these diseases, including bacterial coldwater fish disease, viral haemorrhagic septicaemia, and rainbow trout fry syndrome, the fishes are especially susceptible when their immune systems are already compromised by things such as low dissolved oxygen levels or high ammonia in the water. And in fact, these are conditions that are much more common in aquaculture scenarios than in natural scenarios. Furthermore, since the lifespan of particularly viruses is rather short as a free-floating agent in the water, it's going to have a much higher potential to spread in situations where your population is dense and clumped. Again, such as aquaculture settings.
Susceptible to these viruses and pathogens?
Yes, in fact, a number of the studies that I read focused on, what's the name of it? Fatmouth minnow?
Fathead minnow.
Fathead. Yes, and they showed much higher susceptibility than rainbow trout to the VHS virus.
Interesting.
Up to 50% mortality rates were actually observed in fathead minnows. Now, this varied somewhat due to the nature of the way they were infected with the virus. Now, in the study in particular that I'm talking about, both rainbow trout and fathead minnows were infected, and they're infected in several groups. Some groups were infected by waterborne virus, so the virus was free-floating in the water, and they were infected by circulating water. And this showed much lower rates of mortality than fish that were infected intraperitoneally. Now, our audience may be wondering, does Scott really know the definition of the word intraperitoneal?
I was going to ask.
Yeah, in fact I do. It refers to the injection into the body cavity. And in a video that I will spare you of on YouTube that I saw, the video showed the intraperitoneal injection of a mouse by holding it by the scruff of its neck and ramming a big needle up under its rib cage. It looked very uncomfortable. Now, intraperitoneal injection in this study showed to have much higher mortality rates, obviously in the fish, because they're pretty much guaranteed to be infected because it's injected straight into their body.
I suppose so.
And this gets, again, back to what I was talking about clumped fish populations. And another group of fish that, or life stage of fish, I should say, that are particularly vulnerable to pathogens, including, obviously, the juvenile trout syndrome, as well as the coldwater disease of fishes, is young fish have less resistance to pathogens. Young fish are also, I believe, in most cases, born in very clumped population in spawning beds.
Good point, good point.
So if juveniles are infected, they have a very high potential to infect other juveniles who are in very close proximity to them.
Interesting. Now I wonder, in one of my food webs that I was looking at with Lake Ontario, I was looking, bait fish like alewives or rainbow smelt would be eliminated due to one of these pathogens, and you could see the additional pressure that would be placed from some of the higher trophic levels upon one or possibly no remaining species of that trophic level, and therefore the pressure would be placed lower down in the trophic chain.
Had some pretty high mortality rates in a lot of fish species, but these were experimental scenarios where in many cases the fish were injected intraperitoneally with the virus. In nature, you could expect, because there's a lot more space between fishes, you're going to have lower rates of infection and lower rates of mortality. But still, when you consider other stressors that may already be on that lake, such as low dissolved oxygen due to nutrient loading and eutrophication, toxic mineral loading or toxic chemical loading, warming of waters due to climate change,
sediment transport,
overfishing, any of these factors that are already stressing a population or community would then only be further stressed by the introduction of one of these pathogens.
So I guess what you're saying is that overall these pathogens may not be able to wipe out an entire food web, but likely could be an added stressor to an already overstressed ecosystem?
Exactly. And as we know, when you're dealing with the loss of biodiversity and just about anything in environmental, all of these things are cumulative. And over time, our food webs and our ecosystems are getting weaker. They're getting less resilience
To recover from.
an impact or a disturbance.
Right. Well, good. I'm glad talking to you today. Thank you
that
for the
was nice.
discussion.
Very educational.
Great. Thank you.
Thank you, Jeremy.
Thank you.